So our next unit is one of my favorites, and students usually like it a lot too. It's probably the one that it's probably the one where we change the way that you think about stuff in biology the most. So for example, when you do cellular respiration, it might just be like, hey, you didn't really change the way that I knew stuff from cellular respiration. You just gave me like more details. Like I didn't know that this happened or whatever. But this is one of those ones where you're like, oh, the way that I thought I understood how DNA and genetics and heredity works, I thought it was like this. It's actually not like that. It's actually like quite a bit different than I thought the way it worked. It's not just, most of this class is like, hey, what you learned in ninth grade, but now we zoom in and we look at like more details and it's more complicated, but it's still not like it still kind of works the same way. This is the one where you kind of like have like a frame shift in your mind, more so than other units. So we start off, and at the very top of your notes, there's a little bit of like DNA history stuff. And the DNA history stuff is interesting because it's just interesting. It's a good model of like how science works and how like people build on each other's information and everything else. The other thing that's surprising about it is how new all of this is. So I know 1800 seems like forever ago, but in like the mid 1800s, if I remember right, there's this guy named Hammerling, and at that point we had no idea how heredity worked. So he took these things that look like multicellular things. They almost look like mushrooms or plants, but that's an individual cell. So kind of like a nerve cell, kind of is like long and skinny like that. Not all nerve cells, but like motor nerve cells. If I look really close right there, I can see kind of a dark green oval, and that dark green oval is the nucleus. So that is all one cell. It's not like roots and a trunk and, you know, something like that. So this is all one cell. This is all one cell. And so what he did was he did almost like a graft, kind of like a skin graft. And so he took this middle part of we'll call this one burnt orange, and this one green, and he cut it into thirds. And he took the bottom part of the green one, and he grafted onto it the middle part of the orange one. And then what he expected to grow out the top was orange, but instead green grew out of it. He took this middle green part, put it on this burnt orange bottom. He expected a green top to grow out of it, but it didn't. A burnt orange top grew out of it. And so as he kept doing this experiment with bigger and bigger and bigger and bigger and bigger pieces, it kept working until he cut through the nucleus and the cell died. So what he concluded is like, whatever causes heredity, I think it's in the nucleus. All right, time goes on. Griffith and Avery do what I call 3 no-duh experiments, and then they do one that's interesting. So they start off, and they take a mouse, and they expose it to a heat-killed pathogenic bacteria. What's a pathogen, or what's pathogenic mean? Yeah, harmful or can cause disease. So are there bacteria that are harmless to us? Sure. So those are non-pathogenic. But then like E. coli or salmonella, that might cause food poisoning. So those are pathogenic. So let's say that I kill some pathogenic bacteria, some Streptococcus pneumoniae, and I kill the bacteria and I inject the mouse with it. The mouse is fine. No duh. That's how cooking food works. If I eat a handful of raw ground beef, I might get sick. If I take that same raw beef and I cook it on the grill and then eat it, I'm fine because I'm heat killing the bacteria. So no duh. Then they take live pathogenic bacteria and they inject the mouse and he dies like this. And so he dies. And you're like, yeah, that's why we called it pathogenic, because it's harmful, and that harmed him. No duh. Then they took live non-pathogenic. So this is live harmless bacteria. They injected the mouse and he was fine. No duh, that's why we call it harmless. Then they did something interesting. They took this top one that didn't kill the mouse. They took the bottom one that didn't kill the mouse. They injected both of them together into the mouse and he died. And that's interesting. So first off, you can talk with somebody around you if you like. What appears to make the pathogenic strains pathogenic in those pictures up there? What's it seem like the pathogenic one? All right, what do you guys think? Again, this is all just based off of pictures up here. It's nothing super nitty-gritty detail. Huh? Yeah, looks like this one has a layer of something. So what that's representing is a different type of cell wall. If you've had like a, do we still have microbiology at KCAL or something like that? So you might have to gram-stain bacteria. You might make like gram-positive and gram-negative bacteria. And so gram-positive and gram-negative bacteria have like different cell wall configurations. They have a different cell wall configuration. So it looks like that makes me deadly. And if I don't have it, then I'm not deadly. So here's what he figured out. He's like, all right, I've got this, and it's deadly, but it is itself dead. This one is not deadly, and it's alive. When we check the mouse later, all of a sudden, there are live pathogenic strains in there. They do it over and over, so they're certain they're killing this. So they're like, all right, here's what we conclude. When these guys are dead, whatever causes them to be pathogenic is still around and can still be passed on. Somebody else can take it and become deadly. So whatever causes genetics can actually be moved from one organism to another, and it seems like, at least in bacteria, it's still there even after it's dead. Huh? So that's why you're not supposed to, like in the hospital setting, they try to isolate diseases so they don't like give special resistance to other bacteria. Yes. So one thing that they find out later, not from this experiment, but whenever bacteria die, they, not all, but many of them barf out their DNA. So like if I'm a bacteria and I'm dying, my last breath is I just start barfing out plasmids. And if I'm highly resistant to 5 different antibiotics, so let me give you this scenario. Let's say this is a hospital bed. I'm in there and I've got some like not a big deal disease, but it's highly, highly resistant to antibiotics. Maybe it only causes like ear infections. Not a big deal, but it's highly resistant to tons of antibiotics. So I'm in there for something else, and some of these bacteria get on like the armrest. Cool. They come in, they clean it up, but they didn't get this armrest. So there's some, no big deal, they just cause ear infections, bacteria, but they're resistant to like 10 different antibiotics. Now, somebody else comes in. That's Monday. That guy leaves. Tuesday, I come in. I've got some horrible disease, but it's really easy to treat with antibiotics. It's not very resistant. And I don't know, I cough. And now my bacteria mixed with these bacteria. Now I have a really, really bad one that's easy to kill, and one that's not very bad that's hard to kill. But as this one died, it barfed out its resistant genes. This one sucked it up, and so now this one is resistant. It might take decades for these bacteria to build up resistance to like 3 or 4 antibiotics. But if after I build up resistance to 3 or 4 antibiotics and as I die, I throw out DNA that makes you guys antibiotic resistance, you can pick it up and you can become resistant overnight. Just like the first-person shooter games you guys play, if you kill me and I barf out like 50 joules and 10 guns or whatever that took me forever to earn, and you just walk up behind me, you pick it all up in one fell swoop, you now went from like a level 1 player to like a level 10 player. I don't play any of these games, so I probably messed that up, but you get the idea. So it's kind of like that. So he learned that heredity is still there when the thing is dead probably, and it seems like it can move from one organism to another. The next people that come along, they notice that viruses, when they infect bacteria, this is different than viruses that infect eukaryotes, like you and me. The viruses that infect bacteria usually don't go inside the bacteria. Viruses that infect humans, the whole virus almost always goes inside of our cells. But in bacteria, the virus lands on the outside, then it falls off and leaves, and all of a sudden it's like a virus factory in there. So they somehow, without getting into the cell, they somehow got like genetic code to make viruses in there. So they noticed that, and then eventually, there's various kinds of viruses. Yeah, tobacco mosaic. So it ends up making like the leaves look all splotchy, like a mosaic. And so eventually they do some more tests. They radioactively label DNA and proteins and stuff, and they realize the virus lands on the outside. It injects, these are real microscope images. It injects its DNA inside of the cell. And if I come back and check on that cell in a couple of days, this cell is like a vast like virus factory. So then they conclude, they're like, all right, we think, at least with viruses, that DNA is probably the genetic material. Cool. This guy comes along, and his name is Erwin Chargaff, and he notices this. He notices that anytime he analyzes a string of DNA, there's kind of this pattern. Geez. All right, take a moment, talk with somebody around you. What pattern do you see? Now, you guys already know a lot about DNA that they didn't know back then, so you're at an advantage. All right, so what does he notice? And we would do tests on many, many, many pieces of DNA, but he keeps coming up with the same thing. What does he notice? Yeah, basically, however many adenines I have, I've got about the same number of thymines. Why? No idea. However many guanines I have about the same number of cytosines. Why? No idea. I analyze this smaller piece of DNA. It's got 512 adenines. All right? It's probably going to have around 512 thymines. Maybe it's got 200 cytosines. It's going to have about 200 guanines. He had no idea why, but he figured out that however many A's you have about the same number of T's. However many G's you have about the same number of C's. Huh? It's supposed to represent error, just like experiments are hardly ever perfect. So he didn't conclude right away like it is exactly like that. It's just in their data, they're like, it seems like it's about that. Mm-hmm. So that's Erwin Chargaff. This is in the 1950s. All right, so then Linus Pauling comes along, the greatest chemist of all time. He has already won he's already won one Nobel Prize. He'll go on to win a second one. And he thought that DNA was a triple helix, and he thought that it was facing out. And so if Linus Pauling thinks that DNA is a triple helix, DNA is probably a triple helix, so everybody was trying to make the triple helix work. Just like if Albert Einstein says 2 plus 2 is 5, you're like, I don't think it is, but I'm going with Al. If Albert Einstein says 2 plus 2 is 5, I'm going to start with the premise of 2 plus 2 is 5 and then try to disprove it. Because, like, how do I bet against Albert Einstein? You know what I mean? Like, one of those type of, if Stephen Hawking says black holes exist, or this, if he comes up with something that sounds bizarre, like all that dark matter and weird things like that, you're like, I don't know, but, like, if that lady says it, it's probably true. If that guy says it, it's probably true. So Linus Pauling was looking for a triple helix. Everybody else was like, all right, don't bet against Linus Pauling. He wrote the book on bonding called Bonding, and we gave him the Nobel Prize for explaining how bonds work. The book was actually called something like The Chemical Nature of the Bond, but he wrote a book called Bonding and got the Nobel Prize in chemistry for explaining how bonds work. He discovered the hydrogen bond and then explained covalent bonds and ionic bonds. That's like discovering numbers in math. Like, you don't get more basic than that in chemistry, bonding. So anyway, that's Linus Pauling. He thinks it's a triple helix. Rosalind Franklin in the early 50s is working on X-ray crystallography. And with X-ray crystallography, it's weird. You shoot X-rays at a chemical, and then based off of a chemical that you can't see, and then based off of how the X-rays bounce off, you reconstruct what the chemical looked like. So it's a little bit like a bat using echolocation. So right here, if I'm using echolocation, or imagine I'm throwing a tennis ball. I'm blindfolded. but I can still catch it somehow. So if I throw a tennis ball and I'm really close, it'll bounce back quick. So it's like pow, 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 pow. If I'm farther away, it takes longer. So I can tell that that wall is farther away, even though I can't see it. When I throw a tennis ball over here, it's not going to like bounce off at, I don't know, tangent or whatever the heck, something math. And so it's going to bounce off at some sort of angle like that. And when I get to this, it's going to bounce off at a different angle. And so if I really know my math, based off the angle that this like reflects, I can figure out like the exact contour of this. So I throw a bunch of X-rays at DNA, and if it makes this kind of X pattern, then that means it's a double helix. So Rosalind Franklin and her student were doing X-ray diffraction, and this is photo 51. Technically, her student took it, but she's the one that trained him. They're working on them together. It's almost like I'm the master at tiling floors. I show you everything. I show you all my tricks, and other people maybe don't even know the tricks, and I show you, and as we're working on this floor together, you happen to install that tile, and people come in and they start investigating that tile. I still get credit for it. So some dude named, his name was like Ryan or something like that, takes this picture, and but she can interpret it. He's just taking the picture. She's like, do this, do this, do this, do this. He's like, cool. So she's the one that's interpreting it, and she's like, hey, it's a double helix. There were two double helices, and so in the lab, they divided it up between her and a guy named Wilkins. She studied the A form, and he studied the B form. And so they take off, and they're on. She goes to a conference, presents this at a conference. Watson was at the conference. He didn't quite catch what was going on, and that's the end of that part. Later on, he's invited to visit her lab. So her lab boss and Wilkins' lab boss, whatever that dude's name is, we'll just say lab boss Bob, invites Watson to their lab. And while he's there, Wilkins, her co-worker, showed Watson this, and by then, he did know what it meant. He's like, that means it's a double helix, and everybody there in their lab is like, yeah. But then he's like, oh, wait a second. I'm going to go back to my lab, and they just, out of lab stuff, just like in physics, you guys know how we have these metal physics poles that stick up right here? Like I've got one right over there on my chalk tray. If I put a physics pole right here, and you can like build a ramp, or you can put a ring stand and have like a Bunsen burner under it. You guys are looking at me like I'm crazy. Hang on. Oh, see, 7th period, they put gum on it. So you put those there, and then you can like put a ring stand and like have a Bunsen burner. Did you guys never use Bunsen burners in chemistry? Oh, hot plates. Okay, cool. Anyway, and then physics, you might put clamps and like build a ramp and do some sort of like momentum, inertia, force, something. Or you could just stick your gum on it. Anyway, so they build this DNA model, which is like clamps and crap from around the lab. They publish their paper in 1953, and I think this came up in this class. They give Rosalind Franklin a small mention right here. We've also been stimulated by knowledge of the general nature of unpublished experimental results by Wilkins and Franklin. Her results were unpublished because they were published in the same journal. So her results were published like, it might not be 10 pages, but like, if you keep on flipping through the journal, like 10 pages later, her stuff is published that they're referencing. So Watson and Crick would be like, well, we didn't cite her stuff because you can't cite it. It doesn't exist yet. If I'm writing a book and I publish it in 2005, you can't cite my book in 2004 because it doesn't exist yet. And so it was over here. Now, we go on, and Rosalind Franklin dies. And if you're dead, you can't win the Nobel Prize. So they give the Nobel Prize to Watson and Crick and Wilkins, the guy that was her colleague. Now, over time, Watson downplayed Rosalind Franklin's contribution. he didn't really talk about her in like a positive light. He said she was difficult to get along with. That's why she left this one lab. The reason that the data was shown to him by Wilkins was because she was leaving that lab. And then, like 15 years ago or something, Watson comes back in a Scientific American, and he says, yeah, she should have probably gotten the Nobel Prize too. We should have gotten it in biology, and she should have gotten it in chemistry. And then all of this is so recent that Watson died like 3 years ago. So all of this happened like in my dad's lifetime. Like my dad's still alive, he's 70. But like when my dad was born, they didn't know that DNA was a double helix yet. And my dad is your grandparents' age. It's crazy. All right. See y'all later.